This is the white fat tissue stained with H&E. During this histological procedure, the fat is dissolved from the slides. So we only have the cage of the cell, the very thin cytoplasm around the one single droplet of the fat which filled these cells. So the fat isn't anymore within the cells. That's why it looks like an empty ring. It's truly an empty ring. The diameter of these fat cells is about 50 to 100 micrometer. You see that we have slightly different cells here in size, but we don't know whether this smaller cell is truly a smaller cell or it is just cut at the edge of a larger cell. It's not that important. The range is important, that these are large cells, 50 to 100 micrometer in diameter. Fat cells you've seen before in many of our slides. In this case, this is the fatty connective tissue, and this is a cell-rich type of connective tissue. So not only a few cells here and there, but this is a mass of fat cells. It's worth to look at also the blood vessels within this slide. So here, for example, you see an artery and a vein, a muscular artery and a vein. In detail, we will learn about these at the end of the semester. Here you have the smooth muscle layer in the artery, and the inner lining is given by the endothelial cells. You can judge the diameter based on the red blood cells and also based on the fat cells. So the diameter of this blood vessel is about 200 micrometers. Next to it you see a vein. This is a vein with a much less organized structure in the wall. Occasionally you may find also connective tissue strands. This is a loose connective tissue strand within the fat tissue. If you look around, you will also find longitudinal cuts of capillaries along the edge of the fat cells, like here. Sometimes the fat cells, red blood cells, got out from the fat, from the blood vessels. So here you see these scattered red blood cells. And this is here a nice picture about a capillary as it opens into a venule. So this is here a venule, and this is here a capillary. Between the cells you also see many times cross sections of capillaries. These appear as red dots in case they have a red blood cell in them.